Hi guys, so this is a video to show you the Osmosis Lab potatoes in sucrose solutions data analysis piece. So we have to go over the data analysis, and in this case students are going to be able to use mathematical formulas in data analysis. Students are going to be able to use mathematical formulas to our collected data so that we can look for patterns in the data. I'm going to run through all this in class, but what we're really concerned about is part 4, down here in part 4. This is data analysis. This is what we're going to be doing together. So to access my data, click on this link and make a copy of the document. That's what you want to do first. And you can see that I've got my initial and final masses for all of the potatoes in each of my sucrose solutions. Sucrose solution 0 through 1 molar. My initial mass of my potatoes, and then the final mass of my potatoes. And so we need to calculate percent change in mass from initial to final. That's what we need to calculate. And that will be our final dependent variable. So our formula for that is final minus initial divided by initial times 100, because we are trying to calculate a percentage. So this is the formula we are going to use. I'm going to take my initial potato mass, and we can see that in 0 molar, our final potato mass is actually larger. So I would expect a positive change in mass from initial to final when I finally do the calculation here. So we need to be able to know what the actual percent difference is between the two using this formula. So what I'm going to do is I'm going to go to the blank cell. I'm going to enter an equal sign. Equals tells spreadsheet that I'm trying to do a calculation. And I'm going to need some parentheses here, because parentheses help us separate which of the steps we want to do first. So I'm going to put 2 parentheses first, because I want to get final minus initial first. That's what I want to do first. So I'm going to put 2 parentheses, and I'm going to select my final value and subtract from it my initial value. There, I've selected my initial value. And I'm going to close my parentheses, because again, this is what I want to do first. And once I close parentheses, you'll see that it completes the equation. See that? It's gray on both sides, so it's telling you that the equation is finished, and there's the value that we'll calculate. But we're not done yet, because we want to divide that by our initial value. So I'm going to select my initial value, and now I've got the new calculated value. I'm going to close my parentheses, because now we're going to multiply all of this that we calculated by 100, because this is a percentage. So I'm going to do times 100. And it should calculate to 13.47. Now, you're going to want to do this for each additional molarity. And I could take the time to calculate the percent change in mass for each individual cell all the way down to 1 molar, but that would be really tedious. That would be a lot of time. So instead, what I'm going to do is I'm going to select the cell that I've already put the formula in. I'm going to go to that little box and that little square in the lower right-hand corner. I'm going to hover my cursor over it, and my cursor will turn into a plus sign. And then what I can do is I can click that plus sign and drag and select all of the boxes underneath the cell I've already formulated. What this does is it's going to put the formula I used and apply it to each row. There's the formula I used up on the left-hand side, and that's the formula for cells B and C in row 2. But now we can apply it to each additional row for every single other sample that we have, and that's what I've gone ahead and done. So the program helped me to calculate the percent change in mass for each of our 11 samples. And you can see that up until about 0.4 molarity, we had a positive change in mass. But below that, we have a negative change in mass, meaning the potato has lost mass from there and on downward. So we've been able to now calculate our dependent variable that we were interested in. And I did that by having our initial and our final values and finding out what the percent change in mass is. So this is the actual dependent variable we're interested in. Now, I want to highlight, shift and select all of those boxes, because I'm going to copy them over into the next tab. So I'm going to head and highlight all of those, and then right click or command click and copy. I'm going to take those values, the values only, and I'm going to move them over into a spreadsheet that has other trials very, very similar to ours and similar to the data that we collected, so that we can have better statistical analyses of our data. Okay, so this is the next tab on the same sheet. You can see I have trials 2, 3, 4, and 5 already filled out. This is data from last year, and I'm going to put our calculated data into trial 1. I want to right click, and instead of hitting paste, I want to do a paste special, because if I hit paste, this will happen. I only carry over the formulas and there's nothing to calculate. So instead I'm going to hit paste special, paste values only. And once I do that, it'll take the values that we calculated and bring over those values specifically. Now you'll notice that in trials 2, 3, 4, and 5, students last year did not go beyond 0.8 moles. They only went up to 0.8 moles, which means that our trials of 0.9 and 1 are useless to us right now. They're single data points, and we have nothing to compare them to over here. So I'm going to go ahead and actually delete these 2 cells, and you can go ahead and do that as well. And we're just going to worry about molarity 0 through 0.8. And we're going to compare last year's data to our data. You can see last year's data had a lot of agreement, and our data might not necessarily agree quite that well with last year's data. This is for a few reasons. One, we had more time last year to do this work, so the timing of the lab was a little bit different. And 2, we used different potatoes, obviously, than last year. It was a different batch of potatoes. And so the internal osmotic concentrations of this year's potatoes may be different compared to last year's potatoes. So there are some variables that we cannot control for, but that's okay. Trials 1 through trials 5, we're still all done with the same steps and the same equipment. We now need to calculate the average change in mass for all of the samples, all the trials that we've done for this. And so I'm going to use the average percent change in mass from trials 1, 2, 3, 4, and 5 using this formula, average of a range. So I'm going to go up to my cell, I'm going to hit equals, I'm going to type in average, and I'm going to open parentheses. Yep, and then I'm going to select and press and drag my cursor and select the range of values I'm interested in getting the average from. And I will close my parentheses so they know I'm done with my equation, and then I can hit enter. And it's going to calculate the average of 13, 16, 16, 16, 18. And that's about 15.87. And again, I'm going to want to do this for the additional values, but we learned a trick last time where we can hover our cursor over the small square in the corner, click that square with our plus sign cursor, and drag all the way down to the bottom. And now we've got the average of each of the similar ranges for every additional row that I need. So now I've got all of my average percent change in mass values going across each molarity for all 5 trials. We are worried a little bit about the variation in these trials because our data does not necessarily agree super well with the data from the previous year. So what we want to do is we want to calculate standard deviation. This is a statistical test that tells us how far from the mean our data deviates, essentially. And we have an equation to do that, which is STDEV of a range. And so just like last time, I'm going to use the same range that I did for the average. And I'm going to type this into our box with an equal sign before it. So standard dev, STDEV, open parentheses, click and drag to get my range, close parentheses, and now I can hit enter, and it calculates my standard deviation. And so my values are only deviating a small amount, about 1.84 from the mean, and our mean's 15. So we're not deviating all that far from our mean, which is pretty good. I'm going to click and drag, just like we did last time, to calculate standard deviation for others. And as you can see, as we get lower, our deviation starts to increase. Now we're deviating by a factor of 7.5 from our mean, which is a little bit larger. And you can look at sort of the data and see that, yeah, there's a bigger range or bigger variation in the sucrose concentrations that are higher. I'm going to graph this for you later. I'm not going to have you graph this today because standard deviation graphing is a whole nother lesson. But it's good to notice or think about the variation in data so we can see whether there is a lot of agreement or a little bit of agreement for all of the different trials that we've done. Okay, so finally we have our formulas calculated in the way that we want. We have our independent variable, which is our sucrose solution, and our dependent variable, which is our average percent change in mass from all of our trials. And now I need to know what kind of graph I want. So I go to this graphing website. Really, really good one for figuring out what graph I need to use is the data percent that sums up to 100% or a total amount of time. No. So we're going to go to number 2. Are both your manipulated and responding variables quantitative? The answer is yes. So we are going to go to number 3. Is your manipulated variable level continuous? Is it time and years, for example, or clumped into groups? And we do have a continuous variable. It's a gradient from 0 to 1 molar. And so we're going to end up with either a scatter plot or a line graph when we actually plot the information that we have. How do I know the difference between a scatter plot and a line graph? I usually use this down here to see, okay, a line graph consists of a series of plotted points on the grid that are connected together by a line. This is often used over time, but we're not doing something over time, we're doing something over concentrations. And because it's not a change over time, we don't need to link together our data points. So I'm not going to use a line graph. What I'm going to do instead is go for a scatter plot. And our points are plotted on a grid not joined point to point. We could induce a line of best fit, which we will on our graph, but really this is useful in showing if there's a correlation between two variables. These graphs are useful for showing correlation between two variables, and we do want to show a correlation between sucrose concentration and change in mass in our potato. So this is the type of graph that I am going to use, a scatter plot. So how am I going to do that? How are we going to actually make a scatter plot in Google Sheets? Let's go back to Google Sheets. I'm going to select the independent variable column and the dependent variable column at the same time. And I need to select these two rows at the top that tell my viewer what the information is actually showing. So once I have that selected, I will go to Insert, Chart, and the chart wizard will pop up. And it's going to start off by showing us a column chart or a bar graph. We do not want this kind of graph. So we're going to select a different kind of graph, and we said that we wanted a scatter plot. So I'm going to hit scatter plot. Aha! There it is. Beautiful. And so you can see that our individual data points are plotted against our sucrose concentration. So on the left side is our average percent change in mass, and on the bottom is our sucrose concentration from 0 to 0.8 moles. I'm going to change my, make this a little bit bigger so you can see it, and then I'm going to change my title because I don't like the title that they've given me. So I'm going to change my title so that it includes both the, let's see, yeah, so average percent change in mass of potato core samples in sucrose concentrations. So I'm including my sample organism in my title. That's important. And I can bold or center on the right-hand side when I have chart title selected. Okay, what other information might be useful? We can bold or change the text on our x-axis label. We can bold or change the text on our y-axis label. You can do that for aesthetic reasons. We do see a correlation here, some sort of downward trend. So what I want to do is I want to go to Series. And from Series, I'm going to scroll down and add a trend line. And so that will show that that direct correlation. And we see it as a negative direct correlation, that as sucrose increases, the potato loses more and more mass. So an increase in sucrose has a direct but negative effect on the mass of the potato. I want to use that information and that sort of confirmed correlation between our variables. Let me make this a little bit bigger so you can see a little bit better when I put the equation on here. I'm going to show my Rx, so that is, I'll show my equation, so that's the equation of the line, y equals mx plus b, and I'm going to show my Rx. And Rx is closer to 1 means there's a better correlation, so we're almost at 1, so that's a really, really direct correlation. I don't necessarily need that data point to be filled in on the key, but you can leave it filled in if you want. I might actually rename mine. I think I will rename it to account for the fact that each point is showing a different concentration of different mass of potato, average percent mass change of potato. But it's really important that I have this equation. This equation is going to help us understand what this line actually means in terms of the tonicity of our potato. So we can see where we cross the x-axis. We don't actually know the true value of our x-axis crossing point. So we can use our equation y equals mx plus b, equation of the line, to calculate exactly what that value is of where our trend line crosses the x-axis. So this is our line of best fit, right? We are going to identify what we know in the equation. So m is our slope, b is our y-intercept. Y should be 0, right, at our at the point at which we cross the x-axis. Y should be equal to 0. And the value that we're going to try and calculate here is x. So what is x equal to? That's what we're trying to figure out. And so let's identify what our slope is from the equation at the top. Our slope is negative 70.4. Our y-intercept is 13.4. And we need to solve for x. I'm going to solve for x using this equation. I'm going to put negative before my b divided by m, and now I get my y-intercept is 0.19. And if we go back and look at the graph, you can see where the line of best fit intersects the x-intercept is going to be at about 0.19, just before 0.2 molar. So that would be where there is no change in mass of the potato, right? Where it doesn't increase or decrease in mass. Would be at about 0.19 moles. This point at which it crosses the x-axis. And that's about it. That's all we really need to do for our graph. We could customize a few more things. I think I'm going to make my tick marks on my x-axis a little more clear, so I can think about other molar concentration values that aren't necessarily on here in writing. And I think the last customized thing I want to do, the last customized thing I want to do is to go make my trend line a little bit different color, because the blue here matches the blue of the data points. And so I want my trend line to have just a really different color, something that's more clear to the viewer, so that they can see the trend line a little bit clearer. Feel free to play around with your chart as much as you want to make it look like how you want, but that is what I want my data to look like. All done.